Hello everyone, welcome back to Dr. K. Primer video lecture series presenting by Dr. K. Prem, that's me. In today's episode, we'll talk about the lambda phase lytic and lysogenic cycle. As you all aware that the bacteriophage or lambda phase is a complex and well-studied virus. And it's a perfect example of head and tail virus, where you can see this is the head and these are the tail and these are the tail fibers. The head consists of double-stranded linear DNA molecule, and the tail is useful in the penetration of genome into the host, that's a bacteria. The tail fibers makes holes as well as it provides the attachment to the host molecule, host, that's a bacteria. As like any other viral replication, lambda phase multiplication has 5 steps. So that is attachment, penetration, synthesis of nucleic acids and proteins, assembly of them, and release of progeny. So depends on the cycle it favors. If it is undergoing the lytic cycle, the time required to complete is 20 minutes or so. And if it is undergoing the lysogenic cycle, maybe 45 minutes to complete. During this process of lytic or lysogenic cycle, the lambda phase DNA, which is in a linear shape, undergoes the various changes. So when it is in the head of lambda phase, it is in the linear shape. Once it enters into the host, that's a bacteria, it becomes circular. Then initial replication, theta mode, it looks like theta. During the rolling circular mode of replication, it looks like sigma. And the linear, a long strand of DNA molecule is produced. That's a concatamer, means multiple copies of same DNA is there. And again, that concatamers are divided into, again, single units, and they are packed into the heads of the lambda phase. So viral genome changes its shape from entering into the host, then to packing into the heads of the phase. So today we will focus on these events and hope I'll address you in a better way. You see, here the tail fibers have a lysine molecules. They can break down the cell wall. And this tail can involve in the penetration of lambda phase DNA into the bacteria. Once the bacteria phase genome enters into the bacteria, by using the cos sites which are there at the 5' end of the linear DNA, those cos sites are called as cohesive sites. They are in a single strand form and their length is almost 12 bases. These single strand cohesive sites are complementary. Once the linear double-stranded DNA enters into the bacteria, immediately it is circularized. The cohesive sites, cohesive ends of the lambda phase, they form a complementary, they form a complementation, and that complementary, because they are complementary in nature, the hydrogen bonds are formed, and with the help of bacterial ligase, the linear molecule converted into the circular molecule. Circularization of the lambda phase DNA is very, very essential for two things. One is for to undergo the recombination with the bacterial chromosome. The linear one has to convert it to the circular, or to initiate the lytic cycle and initiate the replication. the molecule has to convert it to the circular. The cell favors the lytic cycle or lysogenic cycle. It all depends on the two things. The concentration of CI protein, that's a C1 or CI protein, or lambda repressor protein, that's a CRO. So here, if the cell has more CI protein because of the early transcription of N, C2 and C3, which stabilizes the CI protein, and integration of the circularized lambda chromosome or lambda genome into the bacterial one, that favors the lysogenic cycle. This is a CRO protein, not CI protein. This is a CRO protein and wrongly labeled. This is a CRO protein. This is a CI protein. More concentration or expression of CI leads to the CI protein, and integration of lambda phage genome into the bacterial DNA favors the lysogenic cycle. Integration of lambda phage DNA is determined by the two factors. One is attachment sites. Attachment sites which are there in between J and N gene, and here there is an integration also. These attachment sites have two halves. One is P and other one is P. And the central element called as O. So similar attachment sites are present even in the bacterial chromosome. They are called as BOB. BOB. The O element of lambda phage DNA and the O element of bacterial attachment sites are complementary. With the help of integrase, the lambda phage DNA integrated into the bacterial DNA through these attachment sites. This kind of integration is called as site-specific recombination. Site-specific recombination. Here, integrase and attachment sites are involved in the integration of lambda phage DNA into the bacterial DNA. That kind of integration is called as site-specific recombination. The now cell enters into the lysogenic cycle. So you see, this is a integrated lambda phage DNA is called as prophase because during this stage, viral genes are silent or not expressed. Hence, the lambda phage genome is called as a prophase. And the integration is a reversible process. Reversible process. So again, they can be separated into the lambda phage DNA and bacterial DNA. Under specific conditions like spontaneous, like specific condition means after the thousands of bacterial cell division, the prophase exit from the bacterial DNA. That's called as spontaneous excision. But before that also it can happen whenever the host is undergoing through the stressful condition such as DNA damaging condition like UV rays or H2O2 which causes the bacterial DNA damage, then the prophase exit from the bacterial chromosome. That process is called as induction. That process is called as induction. Then the prophase which is released from the bacterial DNA again circularized by using the cos sites and a ligase. Now the circularized lambda DNA undergoes the lytic cycle, and where CRO protein is more and the C1 or CA protein is less. Right? Now, with the help of O and P proteins, the DNA replication is initiated after identification of the origin of replication, and they undergo the bidirectional replication. So because of the bidirectional replication, the circularized lambda phase DNA, which is undergoing the theta mode of replication, looks like a theta, Greek word theta. Hence, it is called as a theta mode replication also. Once it is undergoing the theta mode of replication, red and gam, they are the genes where they convert the theta mode of replication to the sigma mode, that's a rolling circle replication. In this one, there will be a nick in the one strand, and they will synthesize the long strands of long strands, and then there is a second strand synthesis. So this long DNA molecule with the single unit of genomes, multiple units of genome is called as a concatamers. You see, a long strand DNA molecule with the multiple copies of lambda phase DNA, they are called as concatamers. So now the action of endonuclease, which identifies the cause sites and cleaves as the cause site and releases the single linear lambda phase DNA molecule with the cause sites at either side. Cause sites at either side, at the especially pi prime. Then the cause sites, which are there with the lambda phase DNA, which are required for the packing of lambda phase DNA into the head. Here, the protein synthesis takes place, and they'll assemble to form a heads and tails and tail fibers. Now, the lambda phase DNA with the cause sites are packed into the head, and then they're assembled and to form a viral daughter cell or a viral progeny. You see, viral progeny. Now, the next phase is a release of the viral progeny that requires the breakdown of both cytoplasmic plasma membrane as well as the bacterial cell wall. So the bacterial plasma membrane is degraded by the poolin molecules. They polymerize and forms the holes through which again endolysin molecules goes, and they are myramidases, and they break down the bacterial cell wall and forms the holes like this, and that leads to the lysis of the bacterial cell and release of viral progeny. You see, they form the holes in the cell walls, then leads to the release of viral progeny. So this is all about the lambda phase, lytic, and lysogenic cycles, and the various features of that. As I told you that during the entire process, the lambda DNA is converted to the circle, and it is entered the prophase, again circle and theta and rolling circle, sigma and concatamers, and again leads to the single double strand linear molecule. So this is all about the lambda phase, lytic, and lysogenic cycle. So hope you like this content. If at all, try to subscribe for regular updates. That's a Dr. K PREM-PRIMER. And if you want to share, there is no limitation. You can share with your friends. And if you like it, don't forget to give thumbs up. And if you have any questions, write to me through the comment section. I'll get back to you once I see. Thank you all for listening. See you again. Until then, bye-bye.